Hello students, today we will learn about microbial production of antibiotics. We will discuss in this video. Antibiotics are the chemical substances that can kill microorganisms or inhibit their growth and are therefore used to fight infections in humans or animals. Most of the antibiotics are produced by microorganisms, that is, product of one organism that can kill other organism. Certain semi-synthetic antibiotics are the chemically modified natural antibiotics. Antibiotics have undoubtedly changed the world we live in and have certainly contributed to the increase in the human lifespan. This is mainly due to the fact that several life-threatening infectious diseases could be conveniently cured by administration of antibiotics. Now, let's get some idea about antibiotics in general. First, let's know about the history of antibiotic discovery, how it is discovered. It was in 1928 the scientist named Alexander Fleming made an accidental discovery that the fungus Penicillium notatum produced a compound, that is penicillin, that selectively killed a wide range of bacteria without adversely affecting the host cells. There are records that in some parts of Europe in 1908 extracts of moldy bread were applied to wounds or abrasions to prevent infections. Although the biochemical basis was not known, the penicillin discovery of Fleming has revolutionized antibiotic research. Now let's know about the wide range of antibiotics. Antibiotics are the most important class of pharmaceuticals produced by microbial biological processes. They are the products of secondary metabolism. Around 10,000 different antibiotics are known and 200 to 300 new ones are being added each year. Most of these antibiotics are not of commercial importance due to various reasons like toxicity, effectiveness, or high cost of production. There are around 50 antibiotics which are most widely used. Now let's know about broad spectrum antibiotics. They can control the growth of several unrelated organisms, example tetracyclines, chloramphenicol. Now let's know about the narrow spectrum antibiotics. Narrow spectrum antibiotics are effective against selected species of bacteria, example penicillin and streptomycin. Now let's know about the microorganisms producing antibiotics. A great majority of antibiotics are produced by actinomycetes, particularly of the genus Streptomyces, example tetracyclines, actinomycin D. The bacteria other than actinomyces also produce certain antibiotics like bacitracin. Among the fungi, the two groups like Aspergillosis and Monilias are important for antibiotic production, example penicillin, cephalosporin, griseofulvin. Now let's know about the application of antibiotics. Antibiotics are particularly important as antimicrobial agents for chemotherapy. A large number of bacterial diseases have been brought under control by use of antibiotics. These include pneumonia, cholera, tuberculosis and leprosy. The antifungal antibiotic griseofulvin has controlled the debilitating fungal skin disease such as ringworm. Now let's know about anti-tumor antibiotics. There are a selected few antibiotics that are in use for control of cancer growth, although with a limited success. Actinomycin D, mitomycin C. Now let's know about the food preservative antibiotics. Certain antibiotics are used in canning industry. Example, chlortetracycline, and for preservation of fish, meat and poultry. Example, niacin, etc. The use of antibiotics in food preservation is usually under the control of governments. Now, let's know about antibiotics used in animal feed and veterinary medicine. Till some time ago, antibiotics like penicillin, tetracyclines, erythromycins were very widely used in processing of animal feeds. Such an indiscriminate use resulted in the development of antibiotic resistance in animals and humans. A new class of antibiotics have been developed for specific use in animal feed. Example, enduracin, tylosin, etc. Likewise, specific antibiotics have been developed for exclusive use in veterinary medicine. Example, hygromycin B, thiostrepton, salinomycin. Now let's know about the antibiotics for control of plant diseases. In recent years, several antibiotics have been developed for exclusive use to control plant diseases. Example, blasticidin, tetranactin, polioxin. Now let's know about antibiotics as tools in molecular biology. Some of the antibiotics can selectively inhibit certain biological reactions at the molecular level. These antibiotics do in fact serve as tools for exploring the knowledge of life science. Thus, certain antibiotics have been used to obtain some important information on DNA replication, transcription and translation. Now let's know about the production of antibiotics, a major pharmaceutical industry. The commercial production of antibiotics is a highly profitable industry worldwide. Annual sales of antibiotics will run into several billions of dollars with an annual growth potential of about 10%. Antibiotics may be produced by microbial fermentation or chemical synthesis or a combination of both. For certain antibiotics, the basic molecule is produced by fermentation and its therapeutic value can be increased by chemical modifications. The cost involved in production and chemical modifications, besides the efficacy of the antibiotics, is very important in its manufacture. Biotechnologists continue their effort to increase the fermentation yield and recovery processes to produce pure antibiotics. Now let's know about some antibiotics. First is penicillin. Penicillins are a group of beta-lactam containing bactericidal antibiotics. Being the first among the antibiotics to be discovered, penicillins are historically important. The basic structure of all the penicillin consists of a lactam ring and a thiazolidine ring fused together to form 6-amino-penicillinic acid. Now, let's know about action of penicillins. Natural penicillins. Natural penicillins like penicillins B and G are effective against several gram-positive bacteria. They inhibit the bacterial cell wall, that is peptoglycan synthesis, and cause cell death. Some persons are allergic to penicillin. Natural penicillins are ineffective against microorganisms that produce beta-lactamase, since this enzyme can hydrolyze penicillins. Example, Staphylococcus aureus. Several semi-synthetic penicillin that are resistant to beta-lactamase have been developed and successfully used against a large number of gram-negative bacteria. Clozaxi- clo- Clozacillin, ampicillin, floxacillin, and azilocillin are some of the examples of semi-synthetic penicillins. These are quite comparable in action to cephalosporins. From the huge quantities of penicillins produced by fermentation, about 40% are used for human health, 15% for animal healthcare, and 45% for the preparation of semi-synthetic penicillins. Now let's know about the organism for penicillin production. In the early days, Penicillium notatum was used for the large-scale production of penicillins. Currently, penicillin chrysogenum and its improved mutant strains are preferred. Previously, the penicillin production used to be less than 2 units per ml, and with the new strains, the production runs into several thousands of units per ml. One of the high-yielding strains is Q176 is preferred by several penicillin manufacturers. Now let's know about genetic engineering for improved penicillin production. Some of the genes involved in penicillin biosynthesis by P. chrysogenum have been identified. Genetic manipulations were carried out so as to substantially increase the penicillin production. For instance, extra genes coding for the enzyme cyclase and acyl transferase have been inserted into C. chrysogenum. Now let's know about biosynthesis of penicillin. L-alpha amino acid. Let's know about the biosynthesis of penicillin. L-alpha amino adipic acid combines with L-cysteine and then with L-valine to form a tripeptide, namely alpha L amino adipyl cysteine valine. This compound undergoes cyclization to form isopenicillin, which reacts with phenylacetyl coenzyme A to produce penicillin G, that is benzyl penicillin. In this reaction, amino adipic acid gets exchanged with phenylacetic acid. Now let's know about the regulation of biosynthesis. Some of the biochemical reactions for the synthesis of penicillin and lysine are common. Thus, L-alpha amino adipic acid is a common intermediate for the synthesis of penicillin and lysine. The availability of amino adipic acid plays a significant role in regulating the synthesis of penicillin. Penicillin biosynthesis is inhibited by glucose through catabolite repression. For this reason, penicillin was produced by a slowly degraded sugar like lactose. The concentration of phosphate and ammonia also influence penicillin synthesis. Now let's know about the production process of penicillin. The lyophilized culture of spores is cultivated for inoculum development, which is transferred to pre-fermenter and then fermenter. Now, penicillin production is an anaerobic process and therefore a continuous supply of oxygen to the growing culture is very essential. The required aeration rate is 0.5 to 1.0 ppm. The pH is maintained around 6.5 and the optimal temperature is in the range of 25 to 27 degrees centigrade. Penicillin production is usually carried out by submerged processes. The medium used for fermentation consists of corn stepe liquor and carbon sources. An addition of yeast an addition of yeast extract, soy meal, or whey is done for a good supply of nitrogen. Sometimes ammonium sulfate is added for the supply of nitrogen. Phenyl acetic acid, which serves as a precursor for penicillin biosynthesis, is continuously fed. Further continuous feeding of sugar is advantageous for a good yield of penicillin. It is estimated that approximately 10% of the metabolized carbon contributes to penicillin production, while 65% is utilized towards energy supply and 25% for growth of the organism. The efficiency of a penicillin production can be optimized by adequate supply of carbon source. Thus, by adding glucose and acetic acid, the yield can be increased by about 25%. For efficient synthesis of penicillin, the growth of the organism formed spores must be in a loose form and not as pellets. The growth phase is around 40 hours with a doubling time of 6 to 8 hours. After the growth phase is stabilized, the penicillin production exponentially increases with appropriate culture conditions. The penicillin production phase can be extended to 150 to 180 hours. Now let's know about recovery of penicillin. As the fermentation is complete, the broth containing about 1% penicillin is processed for extraction. The mycelium is removed by filtration. Penicillin is recovered by solvent N-butyl acetate or methyl ketone extraction at low temperature and acidic pH that may be less than 3.0. By this way, the chemical and enzymatic degradations of penicillin can be minimized. The penicillin containing solvent is treated with activated carbon to remove impurities and pigments. Penicillin can be recovered by adding potassium or sodium acetate. The potassium or sodium salts of penicillin can be further processed to remove impurities. The yield of penicillin is around 90%. As the water is totally removed, penicillin salts can be crystallized and dried under required pressure. This can be then processed to finally produce the pharmaceutical dosage form. Penicillin G and H are the fermented products obtained from fungus Penicillium rhizogenum. Now let's know about the production of 6-amino-penicillinic acid. The penicillins G and H are mostly used as the starting material for the production of several synthetic penicillins containing the basic nucleus, namely 6-amino-penicillinic acid, that is 6-APA. About 10 years ago, only chemical methods were available for hydrolysis of penicillins to produce 6-APA. Nowadays, enzymatic methods are preferred. Immobilized penicillin amidases enzymes have been developed for specific hydrolysis of penicillin G and penicillin B. Penicillin salt of either G or B can be used for hydrolysis by immobilized enzyme system. The pH during hydrolysis is kept around 7 to 8 and the product 6-APA can be recovered by bringing down the pH to 4. At pH 4, 6-amino-penicillinic acid gets precipitated almost completely in the presence of water immiscible solvent. In general, the enzymatic hydrolysis is more efficient for penicillin V than for penicillin G. However, penicillin G is a more versatile compound as it is required for ring expansions. Now, let's know about the cephalosporins. The pharmaceutical uses of penicillins are associated with allergic reactions in some individuals. To overcome these allergic problems, cephalosporins were developed. They have improved stability against beta-lactamases and are more active against gram-negative bacteria. Cephalosporins are broad-spectrum antibiotics with low toxicity. The structure of different cephalosporins we can discuss now. Basically, cephalosporins have a beta-lactam ring fused with a dihydrothiazine ring. Now, let's know about the organisms for cephalosporin production. Cephalosporin C was first discovered in the cultures of fungus Cephalosporium acrimonium, and this organism continues to be used even today. The other organisms employed for cephalosporin production are Emericellopsis species, Pycilomyces species, and streptomycin species. Several mutants of C. acrimonium have been developed for improved production of cephalosporin. Mutants with defective sulfur metabolism or those with resistance to sulfur analogues have high yielding capacity. Certain regulatory genes of cephalosporin biosynthesis have been cloned and genetic manipulations carried out for increased production of cephalosporins. Now let's know about biosynthesis of cephalosporin. The early stages of the biosynthetic pathway for cephalosporin are the same as that for penicillin synthesis. As the tripeptide is formed, it undergoes cyclization to produce isopenicillin N. By the action of epimerase, penicillin N is formed from isopenicillin N. Then penicillin N gets converted to cephalosporin C by a third stage reaction catalyzed by three distinct enzymes, namely expandase, hydrolase, and acetyltransferase. Now let's know about regulation of biosynthesis. A low concentration of lysine promotes cephalosporin synthesis. The inhibitory effect of lysine at high concentration can be overcome by adding L-amino adipic acid. The carbon sources that get rapidly degraded reduce cephalosporin production. Methionine promotes cephalosporin synthesis in C. acrimonium but has no influence on streptomyces. Now let's know about the production process of cephalosporin. The fermentation process concerned with the production of cephalosporin is similar to that of penicillin. The culture media consists of corn stepe liquor and soya flour based media in a continuous feeding system. The other ingredient of the medium includes sucrose, glucose, and ammonium salts. Methionine is added as a source of sulfur. The fermentation is carried out at temperature 25 degree to 28 degree centigrade and pH 6 to 7. The growth of the microorganisms substantially increases with good oxygen supply, although during production phase oxygen consumption declines. Cephalosporin C from the culture broth can be recovered by ion exchange reagents and by using column chromatography. Cephalosporin C can be precipitated as zinc, sodium, and potassium salt and isolated. Now let's know about the chemical synthesis of cephalosporin. In recent years, by using penicillin V as the starting material, chemical synthesis of cephalosporin has become possible. This is being done due to low cost of production of penicillin. Now let's know about the production of 7-aminocephalosporanic acid. Let's know about production of 7-aminocephalosporanic acid. 7-aminocephalosporanic acid is the nuclear structure present in all the cephalosporins. Cephalosporin C produced by fermentation can be subjected to chemical hydrolysis to form 7-ACA. ACA is 7-ACA is 7-aminocephalosporonic acid. This is in short 7-ACA. This is tedious and is associated with several drawbacks. Recently, enzymatic hydrolysis of cephalosporin C to 7-ACA has been developed. This is mainly carried out by two enzymes, D-amino acid oxidase and glutaryl amidase. Biotechnologists have been successful in immobilizing these two enzymes for efficient and large-scale manufacture of 7-ACA. Now let's know about the new beta-lactam technology for production of 7-ACA. Scientists have been successful in producing 7-aminocephalosporonic acid by P-chrysogenome fermentation. This is possible through genetic manipulations. Penicillin N is substrate for the enzyme expandase. Adipilyl 6-aminopenicillinic acid, which resembles in structure with penicillin N, Penicillin N is substrate for the enzyme expandase. Adipilyl 6-aminopenicillinic acid, which resembles in structure with penicillin N, can also serve as a substrate, for example, as a substrate for expandase. By inserting expandase and hydroxylase gene and acetyl transferase gene from S. clavuligerus into P. chrysogenome, the production of adipilyl 7-ACA has become possible. Further, the genes responsible for the enzyme D-amino acid oxidase have also been inserted into P. cryogenome. Both these enzymes act on adipilyl 7-ACA to produce 6-aminocephalosporonic acid. Now let's know about aminoglycosides. Aminoglycosides are oligosaccharides, that is carbohydrate, antibiotics. They contain an aminocyclohexanol moiety, which is bound to other amino sugars by glycosidic linkages. More than 100 aminoglycosides are known. Example: streptomycin, neomycin, kanamycin, gentamicin, hygromycin, sisomycin. Aminoglycosides are very potent antibiotics and act against gram-positive and gram-negative bacteria besides mycobacteria. At the molecular level, aminoglycosides bind to 30S ribosome and block protein biosynthesis. Prolonged use of aminoglycosides causes damage to kidneys and hearing impairment. For the treatment of severe and chronic infections, aminoglycosides are the antibiotics of choice. Streptomycin was the first aminoglycoside that was successfully used to treat tuberculosis, that is against Mycobacterium tuberculosis. Usually, aminoglycosides are regarded as reserve antibiotics since resistance may develop easily. Now, let's know about the organism for aminoglycoside production. Recombinant DNA technique have been used to produce hybrid aminoglycosides and for increasing the fermentation yield. Now let's know about the biosynthesis of aminoglycosides. All the ring structure in the molecules of aminoglycosides are ultimately derived from the glucose. Most of the biosynthetic pathways concerns with the formation of at least some aminoglycosides have been elucidated. Now let's know about biosynthesis of streptomycin. More than 30 enzymatic steps have been identified. Glucose 6-phosphate obtained from glucose takes three independent routes to respectively produce streptidine 6-phosphate, L-dehydrostreptose, and N-methylglucosamine. The former two compounds condense to form an intermediate which later combines with methylglucosamine to produce dihydrostreptomycin 6-phosphate. This compound in the next couple of reactions gets converted to streptomycin. Now let's know about regulation of biosynthesis. Very little is known about the regulation of streptomycin synthesis. A compound named as factor A has been isolated from streptomycin producing strain of S. griseus. Factor A promotes streptomycin production. In fact, factor A minus mutant that can synthesize streptomycin have been isolated. They can synthesize streptomycin on adding factor A. The nutrient sources carbohydrates like glucose, ammonia and phosphate also regulate streptomycin production. Now let's know about the production process of streptomycin. The medium used for streptomycin usually consists of soy meal or soy flour or corn syrup that can supply glucose at a slow rate. The amylase activity is poor in streptomyces species. The initial supply of nitrogen, NH3 and phosphate is also obtained from soy meal. This is required since glucose, ammonia and phosphate is high quantities inhibit streptomycin synthesis. The fermentation conditions for optimal production of streptomycin are temperature up to 27 to 30 degrees centigrade, pH within 6.5 to 7.5, aeration rate is 0.5 to 1.0 ppm. The duration of fermentation process depends on the strain used and is between 6 to 8 days. Now let's know about the recovery of streptomycin. Streptomycin or other aminoglycosides are basic in nature. They can be recovered by weak cation exchange resin in an ion exchange column. Treatment with activated carbon is often necessary to remove impurities. Streptomycin can be precipitated in the form of sulfate salt. Now let's know about tetracyclines. Tetracyclines are broad-spectrum antibiotics with widespread medical use. They are effective against gram-positive and gram-negative bacteria, besides other organisms like mycoplasmas, chlamydiasis, and rickettsia. Tetracyclines are used to combat stomach ulcers. They are the most commonly used antibiotics next to cephalosporins and penicillins. Tetracyclines inhibit protein biosynthesis by blocking the binding of amino acid tRNA to ribosomes A site. That is, in A site, the basic structure of tetracyclines is composed of naphthacene ring. The substituent groups of common tetracyclines we will now discuss. Among these, chlorotetracycline and oxytetracycline are most commonly used in the treatment of human and veterinary diseases besides the preservation of fish, meat and poultry. Now let's know about the organisms for tetracycline production. The first tetracycline antibiotic that was isolated was chlorotetracycline from the cultures of Streptomyces aureofaciens in 1945. There are at least 20 Streptomyces identified now that usually produce a mixture of tetracyclines. High yielding strains of S. aureofacins and S. rimosus have been developed by using ultraviolet radiation and or other mutagens. Such strains are very efficient for the production of chlorotetracycline. Further genetically engineered strains of S. rimosus have been developed for increased synthesis of oxytetracycline. Now let's know about biosynthesis of tetracyclines. The pathway for the biosynthesis of tetracyclines is very complex. There are at least 72 intermediates formed during the course of chlorotetracycline biosynthesis. Some of them have not been fully characterized. Now let's know about polyketide antibiotic synthesis. The term polyketide refers to a group of antibiotics that are synthesized by successive condensation of small carboxylic acids such as acetate, butyrate, propionate, and malonate. The synthesis of polyketide antibiotics is comparable to that of long-chain fatty acids. That is, the carbon chain grows by cyclic condensation process. The synthesis of tetracycline is a very good example of polyketide antibiotic synthesis. As glucose gets oxidized, it forms acetyl coenzyme A and then malonyl coenzyme A. On transmission, the latter gives malonoyl coenzyme A. The enzyme anthracene synthase complex binds to malonoyl coenzyme A and brings out the condensation of 8 molecules of malonyl coenzyme A to form a polyketide intermediates. These intermediates undergo a series of reactions to finally produce chlorotetracycline. Now let's know about regulation of biosynthesis. Carbohydrate metabolism controls chlorotetracycline synthesis. For most efficient synthesis of the antibiotics, glycolysis has to be substantially low, and the addition of phosphate reduces chlorotetracycline production. Now let's know about the production process of chlorotetracycline. The fermentation medium consists of corn steep liquor, soy flour, or peanut meal for the supply of nitrogen and carbon sources. Continuous feeding of carbohydrate is desirable for good growth of the organism and production of antibiotics. This can be done either by adding of crude carbon sources or by supplying glucose or starch. For more efficient production of chlorotetracycline, the supply of ammonium and phosphate has to be maintained at a low concentration. The ideal fermentation conditions are temperature that is 27 to 30 degrees centigrade, pH is 6.5 to 7.5, aeration is 0.8 to 1.0 ppm. The duration of fermentation is around 4 days. Now let's know about the recovery of chlorotetracycline. At the end of the fermentation, the culture broth is filtered to remove the mycelium. The filtrate is treated with N-butanol or methyl isobutyl ketone in acidic or alkaline condition for extracting the antibiotic. It is then absorbed to activate charcoal to remove the other impurities. Chlorotetracycline is eluted and crystallized. Now let's know about the production of tetracycline, different processes. Let's know about the different processes. The production of tetracycline can be achieved by one or more ways. Let's know about the ways. First method is by chemical treatment of chlorotetracycline. Second is by carrying out fermentation in a Chloride-free culture medium. Third is by employing mutants in which chlorination reaction do not occur. Fourth is by blocking chlorination reaction by the addition of inhibitors. Example, thiourea or 2-thiouracil. Now let's know about the microlids. Microlids are a group of antibiotics with large lactone rings, that is macrocyclic lactone rings. They consist of 12, 14 or 16 membered lactone rings with 1 to 3 sugars linked by glycosidic bonds. The sugars may be 6-deoxyhydroxylase or amino sugars. Erythromycin and oleandomycin are 14-membered microlids, while leomycin and trilomycin are example for 16-membered microlids. Now, erythromycin and its derivative clarithromycin are the most commonly prescribed microlids. They are effective against gram-positive bacteria and are frequently used to kill penicillin-resistant organisms. Clarithromycin is currently used to combat stomach ulcers caused by H. pylori. The macrolides inhibit the protein biosynthesis by binding to 50S ribosome. Pauline microlids is the term applied for very large ring microlids that may contain lactone rings in the range of 26 to 28. Example, nystatin, amphotericin. These Pauline microlids are antifungal. Now let's know about the production of microlids. Microlids are produced by actinomycetes. Now let's know about the biosynthesis of erythromycin. In the biosynthesis of erythromycin, the lactone ring are contributed by acetate, propionate or butyrate, while the sugar unit are derived from glucose. Microlid biosynthesis is a complex process and good example of polyketide synthesis, which is analogous to fatty acid biosynthesis. The enzyme lactone synthase is a multi-enzyme complex which is comparable in its structure and function to fatty acid synthase complex. Now let's know about the regulation of biosynthesis. End product inhibition of erythromycin synthesis is well documented. Erythronolide and B inhibits the enzyme lactone synthase. The final product erythromycin has also been shown to inhibit certain enzymes of the pathway. Addition of propanol to the culture medium induces the synthesis of acetyl coenzyme A carboxylase and almost doubles the production of erythromycin. Now let's know about the production process of erythromycin. Industrial production of erythromycin is carried out by aerobic submerged fermentation. The culture medium mainly consists of soy meal or corn steep liquor, glucose, yeast extract and ammonium sulfate. Fermentation is carried out at 30 to 34 degrees centigrade for about 3 to 7 days. Conventional methods are used for the recovery and purification of erythromycin. Now let's know about the aromatic antibiotics. The antibiotics with aromatic rings in their structure are regarded as aromatic antibiotics. In a strict sense, all the antibiotics containing aromatic nuclei should be considered in this group. However, most authors prefer to treat three important antibiotics, namely chloramphenicol, griseofludimus fulvin, and novobiocin in the category of aromatic antibiotics, and the same is done. Same is done. So we'll discuss about these three antibiotics. First is chloramphenicol. Chloramphenicol is a broad spectrum antibiotic that can act against gram-positive and gram-negative bacteria besides rickettsia, actinomycetes and chlamydiasis. However, administration of chloramphenicol is associated with side effects, and the most significant being damage to bone marrow. As such, chloramphenicol is treated as a reserve antibiotic and selectively used. Chloramphenicol binds to 50S ribosomal subunits, protein subunit, and blocks, that is peptidyl transferase reaction, protein biosynthesis. Now let's know about the production of chloramphenicol. Chloramphenicol can be produced by streptomyces, venezuelae, and S. homocysteine. However, chemical biosynthesis is mostly preferred for the commercial production of chloramphenicol. Now let's know about griseofulvin. Griseofulvin is an antibiotic that acts specifically on fungi with chitinous cell wall. It is used in the treatment of various fungal skin infections. Further, griseofulvin is also employed in the treatment of plant diseases caused by biotritis and Alternaria solani. Although the exact mechanism of actions of griseofulvin is not known, it is believed that chitin biosynthesis is adversely affected. Now let's know about the production of griseofulvin. Commercial production of griseofulvin is carried out by employing Penicillium patulum. The chemical synthesis is less frequently used due to high cost. The fermentation is carried out by an anaerobic. The fermentation is carried out by an aerobic submerged process with a glucose-rich medium. Nitrogen is supplied by sodium nitrate. The optimal conditions for fermentation are temperature 23 to 26 degree, pH 6.8 to 7.3, aeration 0.8 to 1 ppm, and the period is 7 to 10 days. Now let's know about nucleoside antibiotics. There are several antibiotics which have nucleoside-like structure. Example: puromycin, blasticidin. S nucleoside antibiotics have diverse structures and biological activities. Puromycin is used to understand the ribosomal function in protein biosynthesis. Neplanosine process antiviral activity. Blasticidin S is a fungicide antibiotic used in plant pathology. Now let's know about the production of nucleoside antibiotics. Let's know about some examples. Puromycin, this is due to streptomyces albonizer. Next is neplamocin A, that is due to Ampullaria regularis. Blasticidin S, that is S griseochromosine. Next is polioxins. This is due to S cacaoi. Okay. Now let's know about the genetic manipulations of streptomyces. The great majority of antibiotics are produced by streptomyces species, a gram-positive bacteria. Of course, there are some other bacteria, both gram-positive and gram-negative, and fungi that can also produce antibiotics. Genetic manipulations in streptomyces have been extensively carried out to enhance the yield of antibiotics and reduce cost of production, besides developing newer or more effective antibiotics. Now let's know about the transformation of streptomyces. Streptomyces strain exist as aggregates of mycelial filaments and not as individual cells. This is in contrast to E. coli. It is therefore essential that the cell walls of streptomyces are broken to release protoplasts for transformation. By adding the desired DNA and polyethylene glycol, the cells can be transformed. These protoplasts are grown on a solid medium to regenerate the cell walls. The transformed cells with desired properties can be isolated for further use. Now let's know about the cloning of antibiotic biosynthesis genes. In general, biosynthesis of antibiotics involves several reactions and participation of a large number of enzymes. It is rather difficult to clone so many genes. Mutant strains of streptomyces that totally lack the synthesizing machinery for a particular antibiotic are very useful. Genes from a clone bank can be incorporated into such mutants and screened for the desired properties. Since this is a lengthy and tedious procedure but has been successfully used for the improved production of certain antibiotics, example undyl protegiocin. Now let's know about direct strategies of gene cloning. It is often possible to identify one or a few important enzymes in the synthesis of antibiotics. From the sequence of amino acids in the enzyme, gene can be constructed and cloned. For instance, the genes for isopenicillin N synthase from P. cryogenum has been successfully constructed in this fashion and used for increased production of penicillins and cephalosporins. Now let's know about genetic engineering for the production of novel antibiotics. Genetic manipulations can be done in the antibiotic synthesizing organisms to ultimately produce totally new and novel antibiotics. Wild type streptomyces coelioli color genes encode the enzymes to produce the antibiotic actinorhodin. S. violaceo rubber produces a related antibiotic, namely granatine. Genetic manipulations can be done between these organisms to produce novel hybrid antibiotics such as medarhodin A and dihydrogranatihordin. The newly synthesized antibiotics are in fact structural variants of the existing antibiotics. As the biosynthetic pathways for antibiotic production and their corresponding genes are better understood, it becomes possible to design newer antibiotics with more efficient action. Now let's know about genetic engineering for improving antibiotic production. For aerobic microorganisms, there is often limitation of oxygen supply that hinders antibiotic production. Some workers have isolated a hemoglobin-like protein produced by the bacterium Vitriosilla species. The gene synthesizing this protein was isolated and cloned in a plasmid vector. The hemoglobin gene of Vitriosilla species was finally incorporated into streptomyces. These new transformed strains have better capacity to take oxygen from the medium even at low concentration. The new strain of S. colicolor was found to produce 10 times more antibiotic actinorhodin than the wild strain, even at a low concentration of oxygen. Now let's know about good antibiotic manufacturing practice. Manufacture of antibiotics is highly commercialized due to a heavy demand worldwide. It is mandatory that detailed clinical trials are carried out before considering the manufacture of any antibiotic. More than half of the antibiotics produced are for human use. If it is therefore absolutely essential that each antibiotic produced is safe, consistent, and poses no health complications. Government authorities play a predominant role in regulating the production of antibiotics. These guidelines ensure that correct procedures are followed at each stage of manufacturing the antibiotics. The product produced should be of consistently high quality. Quality of the product should be checked at different stages of manufacture. It is expected that the antibiotic manufactured should be in the purest form, although 100% purity is not practicable. It is mandatory that the impurities, if any, their quantities and their ill effects should be made known. Thank you. Finally, I have finished this chapter. So, any questions regarding this, you can ask me in the comment box, and subscribe my channels to know more updates. Thank you.